And each floor was like a different institute. You had cardiovascular diseases, neurodegenerative diseases, and then HIV immunology. But now they're all different centers for, microbiome studies, genomics. Yeah, you will see. I want to bring you to the fifth floor so we can meet some of the scientists. They're waiting for us.
Okay, you heard that. We are at the Gladstone Institute in San Francisco, and we're going to go upstairs and meet some of the scientists and researchers who are actively working towards an HIV cure. And as Patricia was saying, this is where they kind of like formalized really taking the drug that we know as PrEP today and formally using that as PrEP.
1 million specimens from around the world. The community was amazing. So the trial was run in Brazil, in Thailand, South Africa. Come on, folks. So this is the fifth floor. This is where Melanie's lab is located. And we're going to go into the lab space and we're going to meet the scientists who are going to talk to you a little bit. The equipment corridors and all these freezers contain specimens, reagents. There's tissue culture room. I don't know what they have planned for you. So we're just going to go meet with them and we'll see what they show us. So Martin Delaney Laboratory that we're bringing over to Gladstone so they can get a little experience of what it is to be working in the lab for those of you who have not ever been in the lab. I'm sure there might be some of you who have. I'm Patricia, I'm the Hope Community Engagement Coordinator.
I'm Pauline Samashima from Canada, and I'm doing the Arts Integrated, a part of Hope.
Alicia Schmer, Miami, co-chair of the CAB.
Ebony Gordon, community partner from San Francisco AIDS Foundation.
I'm Raif Darazi. I'm one of the co-chairs for Hope, and I also do a lot of HIV advocacy.
I'm Jen. I'm a postdoc in the Art Lab. Irene. I'm a graduate student in Melanie's lab. So very similar to what Patricia has already said, we're going to take you on a quick tour and also show you a quick experiment that we did to kind of show you some tools that we use in lab to study antivirals against HIV, for example. So yeah, let's get started.
And then,
so this is the virology laboratory at Gladstone. These are the benches where we work at. Everyone has their own bench. So typically when we're working at the bench, we would just be, wearing lab coats and we're just doing experiments at the bench. But there's also a section of the lab that's called BSL2, which stands for Biosafety Level 2. And in the BSL2 space is where we do a lot of cell culture, which is where we're working with human cancer cell lines, which would be a good model to study diseases, including HIV. And it's also where we can infect human cell lines with different pathogens, including HIV, and study HIV pathogenesis, and also test different antiretroviral drugs to see, when we have the drug present, we infect the cells with HIV, does the behavior of the virus change? Does the replication levels change and whatnot? Yeah, we figured we could go show you an experiment that we set up. And we could go through one of the model viruses that we use to study HIV and show you the cells that we've infected. There's cool colors, so it'll be interesting.
No Resident Evil type stuff going on in here?
No. Human cell lines. We want to protect not only ourselves, but also what we're working with from the, whatever pathogens that we are carrying, right? And so we used these biosafety cabinets. Essentially, there's going to be air circulation all the time. And so essentially, hopefully nothing, if you're using it correctly, nothing should come out and nothing should go in at the same time. Typical stuff we have in here, obviously the biosafety cabinets, but we also have incubators. So we keep these incubators is where we grow cells in, and it's kept at 37 degrees, which is our human body temperature with specific percentages of CO2 and also humidity to make sure the cells are happy.
Tree, what's going on in here?
Yeah, so this is a bead bath. You could also find these typically with water. So essentially, when we're working with cells, we don't grow the cells dry. We grow them in what we call media. It's typically like some saline plus a lot of different nutrients with amino acids, salts to make sure the cells are happy. Obviously, we don't want to dump cold media onto these cells because, cells can be sensitive. And so one of the things we have to do before we introduce media onto the cells is to warm them up up, and so this is just keeping the media at, it should be at 37. We use metal beads instead of water to just keep it clean. It's easier to keep clean versus water. Otherwise, we have to disinfect and change the water constantly, whereas the metal beads, like, you don't. We do autoclave the bead. We clean them annually, yeah. And then, we have centrifuges, which essentially spin down cells, whatever you want to spin down. These are some of the tissue culture plates where we grow our cells in, and I'll grab one very soon. But essentially, one of the most important instruments that we have in this room is this microscope. And through this microscope, we're able to see, very small things on a larger scale. And also we could see fluorescent cells, different colored cells as well. Oh. So I'm just going to walk you through the experiment that we did, kind of like a model virus that we're using. So obviously, I think everyone here is aware that HIV has two different stages of infection. One is the latent stage, and one is what we call productive, which is when there's active HIV being transcribed or being generated in this infected cell, and so you're getting the formation of HIV virions that are being released. One of the tools we use in the lab to study, HIV infection is a virus that is missing a couple genes, so it cannot spread infection. It's a one-round replicative virus, so it can only replicate if you're supplying certain other complementary proteins in the cell to it. Otherwise, it cannot work. We call it GKO. Essentially, there are two different fluorophores, or basically two different fluorescent proteins of different color that will be expressed during different stages of infection. So here, you can see the full genome of HIV, and in the NEF gene, we have replaced the NEF gene with the two different fluorophores. One that's called GFP, which will shine green, and the other one is called RFP, which will shine red. And under different replication statuses of HIV, there will be different colors that are being expressed and produced by this virus in the cell. And so when we infect cells, we could then go on a different machine, or you can even see by eye, which is what we're going to be doing today, to see, which cells are, currently latently infected, or they are, actively transcribing HIV and producing virions, is what we're going for.
And that's indicated by the color.
Yes. Yeah. So this is what an incubator looks like, if you ever wondered. There's different racks, and you can grow your cells in like a flask, a plate. And I think we will just have everyone go around. Obviously, as soon as you take it out of its incubator and out of its natural preferred temperature and conditions, it's already unhappy. So I would say within you want to finish your work within the hour, but you can definitely let the cells hang out for longer than that. If you're doing a latent experiment, that's totally fine as well. We just infected them with this virus, and we treated each of the wells with a different amount of latency reversing agent, and so that would be able to skew how many cells in that well will be either latently infected versus actively transcribing HIV, so a productive infection. Okay, so if you look at we have some wells here called control. If you just go through and look, essentially there are no cells that are green or red. And then there are wells up here with different amounts of our latency reversing agent, and you could go through and see that there are different amounts of green and red.
We have the ability to study the viral protein and the host protein interactors because with Nev and Krogan, we've developed what we call the host protein pathogen protein interaction map. Right, and so this is every viral protein is required to help the virus replicate in some way. In some way, it's involved in the life cycle, or it doesn't exist because viruses are efficient. But what that means is they interact with a host protein to basically turn the host cell into a little viral factory. And so a significant portion of our lab spends time trying to figure out how to manipulate those interactions to see if they are druggable targets. There's a theory where you could drug the host side or you could drug the viral side, right? Direct acting antivirals versus host-directed antivirals. And so we're basically using those two interfaces to see what can we toggle back and forth, right? And so long story short, what that means is we spend a lot of time outside actually trying to make the little pieces of DNA that are required to express the proteins in these cells, right? So sometimes it's not beneficial to use the whole virus. Sometimes we just want to know about that one viral protein and that one host protein, right? And so we can make the cells express those two at higher amounts.
Right.
And then you can do cell biology or biochemistry to figure out what that interaction is more like.
We make art while we're trying to learn, and there was one of the pieces that I was working on when we went to a session where it was like where the shape is like matching with another shape. So the corner is matching with the other protein. And I was thinking it's just like a relationship, like two people trying to like
figure it out. Yeah.
Yes.
Absolutely. Absolutely.
And there's so many variables about getting that relationship right. It just seems really complex.
Right, and so that's what you try to do is try to break the host viral protein interaction, right? But then what you then have to make sure is this isn't toxic to the host, right? So the reason like a lot of antiviral research is really difficult is a lot of the things that are required for the virus are also required for us to survive.
Yes.
So you have to be very delicate when you're,
Yeah, that's the thing.
Right.
You know, you have to be very careful about the toxicity of your molecules that you're going to use to try to prevent the virus from either replicating or integrating. Thank you so much for preparing an experiment for all to see. I think that opportunity to, you know, for folks who don't go into the lab to see what it feels like to be, you know, in a cell culture. This is called a BSL-2. We also have a BSL-3 facility, which is the next level up. BSL, biosafety laboratory, and the level, your different levels. You know, in the lab is what called BSL-1. You know, you can cultivate certain bacteria where you're doing like plasmids or, you know, but BSL-2 is already, you know, you have some pathogens in here. You know, you see the sign, the biohazardous, you know. And the BSL-3 has a negative pressure, which means that there's a SAS when you go in, and we can walk by it. There's two doors. So you open one door and you go in a SAS, and then there's another door to go in the actual space that looks like this. That's where you're manipulating things that have a higher risk as a pathogen. If you spill it, for example, with HIV, with certain level of concentration of virus, you have to do it in the BSL-3 versus here in the BSL-2. Cells that are infected can be in BSL-2, whereas, you know, I don't know which viruses you have in the BSL-3 right now, but it used to be there.
Right now, it's only HIV at high concentrations. We have a
At one point, I remember we were working with something else in there.
yeah. Right now, it's just HIV. We have a different BSL-3 for SARS-CoV-2, but this is not. It's not. Yeah, it's not on this floor. Yeah.
And then the next level up is BSL-4, but there's no here at Gladstone.
there's
That's
like
where,
10 in the whole United States.
yeah.
I
That's
remember
where
when
you
they're saying
have the-
because of earthquakes that you couldn't
I
qualify.
mean, you don't want
for
to BSL-4 near a
You don't
phone line.
wanna-
Do accidents happen, or do people get infected?
No.
No, because most of our people are trained well enough that, you know, it basically, all of these lab safety levels, you're trained to design your experiment in a way to take minimum risk.
And you're trained to enter. Before you go into BSL-3, you have to go through a whole training process. And we are trained to clean up spills. We have to go through all these biosafety trainings over and over again in case there was a spill or something. Thank you so much.
Thank you.
But I thought maybe we could go take a look at the BSL-3 facility. So you see, every time you go in, this is like, for example, the equipment room, this is a SAS, this negative pressure. So if there was any spill, it wouldn't. So all of that is SAS. But I spent hours and days in there. And with the IPREX specimens were in there. Oh my God, it makes me emotional. Oh, the light is off, but maybe you can peek through here. So that's where the people go in, they gown up before they can go inside. And so, you know, supposedly you're not opening this door while the door on the other side is open. So you can take it.
Like an airlock.
So, to enter the BSL-3 here, it's by key card access, and obviously, as Patricia's mentioned, you know, there's like training involved.
Want to take a look?
And in this ante room is what we call this. You're, you know, you're putting on like what you've seen doctors wear, like a hospital gown, safety goggles, double gloves, and all of that. And then you go all the way into the actual like laboratory space which will still very much look like a BSL-2, but you're just working and like you have more safety equipment on you, and also the mentality in which you are working will be slightly different. You're more cautious when you're pipetting or like handling the cells or whatnot.